Good morning. Welcome to today's Future Supply Actions Funding Program webinar. I'm Warren Tights. I'm the team manager. I am a team manager within the Water Resource Management Group at Metropolitan and also the project lead for the FSA program. This webinar is being pre-recorded. The recording and the slides will be posted on our website at some point. Next slide, please. I'm just going to go through a brief introduction to Metropolitan and the FSA program. Then we'll do the presentation, and then we'll ask some questions of the presenters when that is done. Next slide, please. So Metropolitan is the primary wholesaler for Southern California. We deliver our whole imported water supplies to 26 member agencies serving 19 million people and supporting a $1 trillion economy. Next slide, please. But over the past 30 years, we've diversified our supplies with tremendous investments in conservation, storage, transfers, and then also, very importantly, local supplies. And innovation in local supplies is fundamental to our success as a region as we advance our One Water approach. We need to build climate change resiliency into our portfolio, and this is where our future supply action funding studies, like this particular study on recharge basins and Quagga mussels, fits in. Next slide, please. So Metropolitan initiated the FSA program in our 2010 Integrated Resources Plan, and with the goal of driving innovation in local supply development. And so through the program, we collaborate with our member agencies like Santa Ana and their sub-agencies like the Orange County Water District to pilot new approaches. Because let's face it, we've got 19 million people that we're responsible for. We've got billions of dollars of infrastructure, and we need to de-risk new technologies before plugging them into our system. And the FSA program is one of the ways that we can pilot and de-risk these new technologies. And so we also want to create, through this program, opportunities for new supply development by removing barriers. They can be technological barriers, institutional barriers, regulatory barriers, and funding barriers. This particular study helps with regulatory barriers and also technological barriers. Okay, so next slide, please. So our current program has funded 14 member agencies, just over $3 million of co-funding provided by Metropolitan, but this supports about $8 million of total regional investment. And we've funded an additional 7 studies with the Water Research Foundation for another just under $1 million. So this particular program is about a $4 million program. Next slide, please. Oh, wait, can you go back one slide? So these studies that we're funding, they're not going to help us through this current drought, which is severe, but they will help us with future droughts and help us build climate resiliency into our portfolio. Okay, so next slide, please. Our member agency partner for this study is the City of Santa Ana, and they work very closely with their partner, Orange County Water District. And our speakers today include Cesar Barrera. He's the Deputy Director of Public Works for the City of Santa Ana. Adam Hutchinson, he's a recharge planning manager with Orange County Water District. And Kelly Stockton-Feedy, she's a senior research biologist with KASF Consulting. And before I turn it over to Cesar, I do need to read a disclaimer that this study investigated a particular product for controlling Quagga mussels. Metropolitan co-funded this study, which was carried out by the project partners I just described. But this is not an endorsement by Metropolitan of this product or the vendor. That said, let me turn it over to Cesar, and Cesar, take it away.
Thank you, Warren. The City of Santa Ana is one of 19 major retailers in Orange County Water District service area, serving a population of about 334,000 residents. As a groundwater producer in the Orange County Water District service region, the city relies on Orange County Water District's management of the Orange County groundwater basin accounts for roughly 75% of our annual production here in Santa Ana. Since quagga mussels were discovered in Lake Mead in 2007, they have spread throughout MWD's Colorado River Aqueduct as far south as Lake Skinner near Temecula. Despite chlorination and other efforts, quagga mussels have infested reservoirs used for capture of local runoff, as well as rivers, streams, and canals used to convey both local supplies and imported supplies to groundwater replenishment facilities. Orange County Water District uses many of the same facilities to recharge the groundwater basin with stormwater and imported water. The presence of quagga mussels has affected the ability to maximize the capture and recharge of local supplies. This affects the amount of water available to those of us that rely on the groundwater basin locally here. For example, Irvine Lake receives both imported Colorado River water and local storm flow. Downstream of Irvine Lake are Orange County Water District Santiago basins, which are used for groundwater replenishment. The deliveries are no longer possible due to the presence of quagga mussels in Irvine Lake and the potential of infestation in Santiago Creek and Santiago basins, which reduces the groundwater availability to cities like Santa Ana. Next slide, please. As a MET member agency, the city took the lead in obtaining grant funding from MWD that covered 50% of related costs. In August 2019, the Santa Ana City Council approved an agreement with MWD and Orange County Water District for the study. The study examines ways to maximize the groundwater basin recharge with imported and local supplies, which will in turn increase available water supplies to the Santa Ana and other suppliers that rely on the groundwater basin. This project is a great example of local collaboration on an issue that affects all California and beyond. It was important to the city of Santa Ana to partner up on this project and bring relief for impacted communities and continue the supply of our award-winning water. Thank you.
Thank you. I'm Adam Hutchinson. I'm the Recharge Planning Manager for Orange County Water District. And I just wanted to introduce the study to you before Kelly gets on and takes over. With limited water resources in Southern California, we rely on imported water, recycled water, and stormwater. And as Cesar mentioned, our groundwater recharge facility is not just operated by us, but other people rely on both imported water and other sources. And so we know that the quagga mussels present in the Colorado River Aqueduct system and have been detected at other state water project sites such as Pyramid Lake, Castaic, and Lake Piru. And one of the issues here also is that the quagga affected water is restricted for groundwater recharge by the California Department of Fish and Wildlife. You have to put together a quagga response plan, and those response plans have to be very specific in terms of how this water is recharged, where it's recharged, and how it's mitigated for. Next slide. So to give you the specifics on Orange County Water District, the panel on the left here shows our entire recharge system located primarily in the cities of Anaheim and Orange. And we have over 1,500 acres of land and over 25 different recharge facilities. And they're shown in light blue. And the star up there is our OC28 connection. That is where we can receive imported water from Metropolitan Water District. There's also a connection upstream not shown where the water can be delivered directly into the Santa Ana River. But because of the presence of the quagga mussel, we are limited to where we can put this water. And the panel on the right, all the recharge basins that are hatched, which is 87% of our system, can no longer receive imported water from the Colorado River because of the quagga mussel. So this is really constraining our ability to recharge Colorado River water, especially if we want to take large quantities in a short period of time. Our entire system used to be able to take this water, but now because of the quagga mussel, we are limited to only those basins shown in light blue. And the reason those basins can be used is because they can be dried out and desiccated and then put back online. And thank you. The Project had widespread support. We thank the city of Santa Ana for their support in this project and Metropolitan for co-funding it. You can see the parties shown in green are parties that have contributed financially to the study, including the Water Replenishment District, United Water Conservation District, for Lake Piru work, the main San Gabriel Water Master, Central Basin Water District. They all share, like OCWD, constraints in using their facilities with Colorado River water because of the Quagga mussel. We've also had support from the California Department of Fish and Wildlife and Department of Water Resources. They are also interested in the outcome of this study to potentially assist them in any issue they may have with the Quagga mussels in their area. As far as implementing the project, our staff, along with Kelly's firm and Tussle Technology, implemented the project that you're about to see. So I want to thank them for all of their hard work, especially with the challenges of COVID. We really had to adjust our sampling schedules and delay sampling by a year to address some of those constraints with COVID. But fortunately, we were able to work through all of that and get a really good data set that we're excited to share with you today. So Kelly, I think it's your baby now.
All right, so the objectives of the study were to evaluate EarthTech QZ for controlling invasive quagga mussels. And we also looked at the toxicity to non-targets. So we wanted to see how well it worked and whether it was safe or not. We looked at a couple of different water qualities within Southern California, Lake Piru, which is up north there, which is state water project water and local runoff. Then we also looked at Lake Matthews, which is the Colorado River Aqueduct Reservoir. And then Weymouth Water Treatment Plant was another sampling site, and it receives Lake Matthews water that has been exposed to chlorine within the pipeline. And that pipeline residence time is about 8 hours. So to give you a little bit more background on quagga mussels, they are invasive species. They reproduce very rapidly. Females can release up to 1 million eggs per spawning event. And in these warm waters, such as Lake Mead, they can have multiple spawning events per year. They are highly efficient filter feeders and they impact the food web. And so they usually eat bacteria and small algae, which then leaves less food for zooplankton and small young of year fish, therefore impacting the food web as it goes up. This picture is a picture of the quagga life cycle. And so I want to point out that these are broadcast spawners. The adults release their sperm and egg into the water. They fertilize and form a little embryo that then becomes a Veliger. And the Veliger is the stage where they have a hard shell and that we can easily pick them up with a plankton net. The size range from about 50 microns to 500 microns. And then they'll grow a foot and settle onto anything hard and grow and become adults. The Veligers are within the water, whereas the adults are in the benthic area. So since Colorado River Aqueduct transports just water, that's what we're concerned about. As Caesar mentioned, quagga mussels were found in Lake Mead in 2007. And I think as mentioned previously as well, the Met does treat the Colorado River Aqueduct using chlorination, desiccation, and physical removal tools. So we wanted to look at EarthTech QZ. It is a labeled molluscicide. It is a copper sulfate pentahydrate as the active ingredient, and it's NSF ANSI 60 certified, which makes permitting easier. Again, we wanted to target the Quagga Veligers because that's what's being delivered to entities like Orange County. And then we wanted to look at the safety of it, the non-target aquatic organisms. We tested 3 different representative organisms. And then we also wanted to keep in mind that the copper limits in drinking water is about 1.0 milligrams per liter. So these are our methods. We went out to each testing location, did a plankton tow with a 50 micron plankton net, filtered it through 500 microns. So we kept anything that was 50 to 500 microns in size. And then we took that and a bunch of water back to the lab and conducted Quagga Veliger testing. We did 2 different temperatures, 15 and 20 degrees C. We looked at 5 different doses of EarthTech QZ, 0, 3, 16.7, 33.4, 50.1 microliters per liter. The associated milligrams per liter as copper is listed. We did 4 exposure times per concentration, so half an hour, 2, 5, and 24 hours. We analyzed them after the exposure time. We stained them and then analyzed them. And after analysis, we put them back into clean water and allowed them to sit for another 24 hours and analyze them again. And so this is the recovery period. And we are presenting just the recovery results in this study. During treatment, we did the quality monitoring. We did testing on the treated water of total and free copper. This allowed us to look for, ensure that our copper was at the correct concentration, as well as the decay rate of copper. We looked at pH, dissolved oxygen, conductivity, and temperature. For the raw water, we looked at dissolved organic carbon, alkalinity, hardness, chemical oxygen demand. And then we sent water to a lab that did acute toxicity testing on the 3 non-targets. So into the results, I want to point out that mortality here on the y-axis, hours treated on the x-axis, and I'm only going to present the 16.7 microliters per liter EarthTech QZ or 1.0 milligrams per liter on this graph. So this is Lake Piru's results for each temperature. It was conducted in December 2019. We achieved 23% and 42% mortality after 24 hours as the recovery period. And then mortality increased with higher concentrations. So we achieved 66.4% at 33.4 microliters per liter EarthTech and 92.5% at 50.1 microliters per liter EarthTech. To build upon this graph, I'm including the Lake Matthews in yellowish colors. We conducted these in September 2020 and July 2021. In 2020, we achieved 99 and 92% mortality at the 16.7 microliters per liter EarthTech QZ after the 24-hour exposure period. In 2021, we achieved 93 and 81% mortality. And then as Lake Piru, mortality increased with higher concentrations. So we achieved 97.3% mortality at 33.4 microliters per liter EarthTech, and then 98.9 microliters per liter at 50.1 EarthTech. To continue, these are Weymouth's water treatment results in the green and gray. And so we conducted these alongside Lake Matthews in September 2020 and July 2021. In 2020, we achieved 100% mortality in all temperatures treated after 24 hours. And in 2021, we achieved 99 and 100% mortality at the 16.7 microliters per liter EarthTech. Mortality increased with higher concentrations again, and we achieved 100% mortality at both the 33.4 and 50.1 microliter per liter EarthTech QZ. And I just want to remind you that Weymouth water is chlorinated Lake Matthews water. So to compare all of the concentrations, all of the doses, we built a time dose variable where the time dose is the exposure time in hours multiplied by the copper concentration in milligrams per liter. And that's represented on the x-axis. And so that way we could combine it all and have one line per site. So Weymouth water treatment plant had the highest mortality at all concentrations over time, the gray line. And Lake Piru had the least mortality over time, and want to point out that that is State Water Project water with local runoff. When we ran this through the linear regression modeling, we found that temperature and year were not a significant factor. So that means that these results are not seasonally variable. So that's good. You can go in and treat anytime you want. However, location was a very significant factor. And so we were trying to figure out why location was a factor. And in the field, we observed that the larger veligers had higher survival rates. So this is a picture of what I see through the microscope. And this is stained. And so the stain stains dead veligers. And there's different size classes within the sample. And as you can see, there's a dead D-shaped here, a dead small umbonal, a dead large umbonal. But we have a very live, large umbonal that's not stained and surviving treatment. So the veligers were, or each sample site was just counted, the size class, sorry, size class was just counted. And I looked at the distribution of the size class of Quagga mussel veligers used in testing. And then I ranked each site by density of large to small. So on the left side of the graph are the sample sites that had the largest veligers, where on the right side of the graph, they had the largest density of small veligers. Locations sampled during the same week were combined because they had similar size class distributions. And then we found that Lake Matthews had the highest variability of size classes. So they had one of the highest densities of large-sized veligers as well as small veligers. So when we break that out, just in this graph, the Lake Matthews is split out. And this is the yellow line is the line that has the largest size class, and this blue line has the smallest size class. We found that the largest size class from Lake Matthews had the lowest mortality, and the smallest size class had the highest mortality. So we came to the conclusion that size class explained variation in mortality at the location, but it didn't explain the variability between locations. So then we looked at water quality, and these are the water quality parameters for each site tested, an average of all testing. And through linear regression modeling, we found that hardness was not a significant variable because all these waters are considered hard. And then alkalinity and dissolved organic carbon were considered significant variables. So water quality explained the differences in mortality between the sampling sites. Now on to acute toxicity of non-targets. We looked at 3 standard non-target organisms, the water flea, rainbow trout, fathead minnow. We sent water that we collected each week at 2 aquatic bioassay and consulting labs, which they performed aquatic toxicity standards per standard EPA test methods. And that's a 96-hour exposure to a constant Earth Tech concentration. This is the result. Again, we combined all the data into that time dose calculation there on the x-axis. so that we could do some comparisons. And so the water flea in yellow was the most sensitive, whereas the fathead minnow was the least sensitive. And these are 96-hour trials. And in the Veliger test, the highest exposure time was 24 hours. So we added then the Quagga mussel graph mortality to this, and we find that Quagga mussel veligers are as sensitive to copper as the water flea in Lake Matthews and Weymouth water treatment plant. But in Lake Piru, the rainbow trout and water flea were going to be impacted before the Quagga mussels were. Fathead minnows would survive an earth tech treatment below 1.0 milligrams per liter. At 3.0 milligrams per liter, they do not survive. And then one caveat to testing non-targets is that they're using sensitive sized organisms or very small. So that may be something to consider when doing an actual treatment. So to apply these results, we can look at doing a treatment at OCWD's OC28 that Adam had a picture of earlier in this presentation. And they receive Lake Matthews water through a pipeline, and it's a similar travel time to the Weymouth water treatment plant. So we can look at Weymouth water treatment plant's results to give us an indication of what we'd achieve at OC28. So if we treated for 2 hours at 1 milligram per liter copper or 16.7 microliters per liter earth tech, we'd achieve 70% mortality. If we were able to treat for 5 hours, then we could achieve greater than 90% mortality given our results. However, site-specific testing needs to be conducted to determine an effective dose, look at the copper decay rate to know what we are releasing downstream, and that would lead into impacts of species of interest. We might need to look at those rather than maybe the generalized ones that we've already looked at. Some other potential applications, as Caesar mentioned, the Water Replenishment District and Main San Gabriel Basin Water Master also gets water and has some of the same problems as OCWD. And so this could be a potential use. Irvine Lake also has Quagga mussels, and they receive local runoff and Colorado River aqueduct water. One thing to point out is that other parts of the country that have used Earth Tech QZ, they use it in closed water bodies, and they have not found harm to other aquatic species. So the fish are still living after a treatment, and the zooplankton communities come back and flourish, and the Quagga mussels are gone. So the lessons learned out of this study was that the main study that people found Earth Tech so promising was a study from Waters et al. In 2013, and she tested water from Lake Mead and found that she could get 100% mortality of the veligers with using 3 microliters per liter Earth Tech in 30 minutes. I've tried to repeat this, and our study tried to repeat this in the Southern California waters, and we could not achieve this, and we could not achieve 100% mortality in any of the water within a 30-minute exposure time. We got about 70% mortality at the highest concentration tested. And then another thing that we learned was that lower doses were safer for non-targets. However, that will take a longer contact time to kill veligers. Some limitations is that this was a bench top testing approach, and it did not provide the full assemblage of organisms within the water column, since we filtered out the water to make it usable. And so Earth Tech QZ's effectiveness could be decreased because it is affected by organic matter. Again, the standard acute toxicity methods were 96 hours with small sensitive organisms versus the Veliger tests, which the highest exposure time was 24 hours. So that limits correlation of the results. And so we need to complete testing on organisms of interest and of ecological value to really define how Earth Tech's going to work in each system. And so to do that, we recommend large-scale field testing approaches such as mesocosms in further additional studies. And so this could be flow-through systems or little tubs with samples of water in large volumes. So in conclusion, Earth Tech is effective. It's dependent on water quality, so hardness, alkalinity, and dissolved organic carbon. Chlorine-treated veligers were more susceptible to an Earth Tech treatment. And so you could, after chlorine treatment, you could use a lower dose of Earth Tech. Larger veligers took longer to be killed than the smaller veligers. And then lower doses are safe for non-targets. However, that will require a longer contact time to kill the quagga mussels. And as I mentioned previously, we recommend site-specific testing for prior to each application so that you know what you're going to get to achieve acceptable veliger mortality, to understand what non-target mortality is going to be, and do these applications with the full planktonic assemblage. With that, we will take any questions.
Great. Thank you, Kelly. Thank you, Adam and Caesar, for a great presentation. This study is going to help any agency with a recharge basin that could be affected by quagga mussels. So very important for our region. Those recharge basins are critical for maintaining our groundwater supplies. And I don't think people realize that our region captures close to 400,000 acre feet of stormwater a year with these recharge basins. And also we use them for recharging recycled water as well, and imported water. So these recharge basins are very important, and this is a great study. We have time for some questions, and why don't we open it up? Don Bentley and myself are going to pretend that we're an audience for this particular recording. So Don, why don't you ask the first question?
Thank you, Warren. Yeah, the first question is for using this in an operating water system, what is the typical application method? Is it something that's a continuous application, or is it periodic as needed?
That's going to depend on your use. And so if you have water flowing constantly, you'll probably want Earth Tech flowing constantly as well. If you are able to hold your water before it's released, then you could do a dose and hold type situation, and then have clean water to release downstream. So it's going to be how's your water running, and if you could stop it and hold it, it'll be a lot cheaper and probably easier to do than a flow through system.
Great. Thank you for that question, Don, and the answer. This next question I have, I think might be for Adam. What would the permitting process look like for using a product like Earth Tech or any other molluscicide on a recharge basin?
Yeah, great question, Warren. Can you hear me?
We can hear you fine, thank you.
Oh, great, thank you. Yeah, that's the reason we brought the California Department of Fish and Wildlife into this project, is because they're the ones that issue the permits for using a source affected by quagga mussel anywhere in California. And so we wanted to bring them in so that if we saw promise in this approach, we would actually work with them to make this a viable approach in the quagga response plan. Currently, right now, we can use the Earth Tech and other methodologies for spot treatment, kind of clean up if there's an infestation somewhere. But this would be more for long-term use. And so I think based on the outcome of the study, we are going to be sharing these results with them and seeing what they think in terms of this being a viable pathway for future application.
Great, thank you. I think Don, you're up next.
Question, are there any similar products to Earth Tech QZ, and any examples of something like this having been tested before?
Earth Tech is one of the more promising chemicals on the radar, I guess, that's labeled and has done the legwork. Has shown success. However, that is not labeled for use, and in our sodium rich waters, it doesn't quite work as well. Here in Southern California and lower Colorado River water. There's some proprietary ones that have shown promise, but they're still in the early stages of research and funding, let alone being labeled for use. So.
Yeah, thank you, Kelly. I'll just add something that I learned through this whole process is in the old days, you probably heard of copper sulfate being used to deal with the algae blooms and whatnot. And from what I've learned is the bioavailability of copper sulfate is relatively low. So you're spending a lot of copper that isn't used and it ends up settling on the bottom and being wasted, if you will. Whereas Earth Tech, my understanding is the way that it's formulated is 100% of the copper is bioavailable. So it's absorbed by the organisms. Any living organism within the water body will absorb this copper. And so it's much more efficient and effective at lower doses. And so I think that's one of the reasons why we thought this was such a promising approach. And like Kelly said, maybe there are other products out there in development, but that's one of the key features of Earth Tech versus just simple copper sulfate and other similar types of products.
Great, thank you. I think this next question is for Kelly. Kelly, you addressed some of this in your presentation, but can you give an example of what sort of the next steps in implementing larger scale field tests would be? You talked about the mesocosms and you had that slide with the basins out. Could you get into that a little bit more detail?
So yeah, as an example, if Adam wanted to go ahead and try and do a treatment, we would probably take over his lab again and pull water directly from where treatment was going to be initiated and pull it into some buckets like the picture that we saw, or some sort of holding basin outside of their system so that we don't treat the Santa Ana River by accident, and put in some Quagga Veligers, do a typical treatment and look at the decay rate of copper over time, look at the Veligers over time, maybe throw in some adults to make sure that we're killing those, and go from there. We would want to look at the zooplankton assemblage and then any species that are of concern that this water would get released onto or are within the treatment system. And so we could either look at, these studies would want to be kind of tailored to how it's actually going to be used in treatment. And so if these organisms are not going to be hit with copper until the very end in the Santa Ana River, we need to know kind of what that decay of copper will be and that uptake of algae over time is going to be. And so it's a bigger study to kind of watch all those variables and see how all those variables come together than just in a beaker.
So would you recommend, so if another agency was going to do a study for the recharge basin, would you recommend them going directly to that approach? Or would they want to start with something like what you did in this study?
We would want to know their water source, because we need to know, since water quality is a factor in treatment and concentration. We need, if it's Colorado River Aqueduct water, sure, we could go forward in those next steps. But if they're getting local runoff like Irvine Lake is, that may change some of those water quality variables. And so we might have to do the benchtop testing just to make sure that we're getting the mortality that we think we want.
Interesting, thank you. I think Don, you're up next.
Yeah, again, this is down the road, and possibly trying to envision what a treatment system would look like. Would this be something like with large tanks of storing the earth tech, and are there any issues associated with delivery and is it like we were talking about earlier about flowing water, and flowing water would be some sort of a metering system to deliver the earth tech to the flowing water? Any thoughts on this?
So I'm not the expert, but I have worked with David Hammond, who is the expert for Earth Tech QZ, and it's very similar to a chlorine treatment. The same equipment as far as metering pumps and all that stuff. You could just take one hose, your chlorine hose out, and put your Earth Tech hose in. As far as logistics and all of that stuff, you'd have to speak with the company. Earth Sciences Incorporated, I believe.
Yeah,
Right, think of
Warren, I'll build on that, Warren. Or Don. Yeah, we kind of brainstormed what that might look like for Orange County Water District, and you saw that picture of our OC28 connection. And that would be an ideal location for a metering system, like Kelly mentioned, to treat whatever portion of that flow we would discharge into the Santa Ana River. It would go down a channel before going into the river. But so we would just treat a side stream of the imported water we're getting and meter in the right dosage at that location. And it would only be for, a few months out of the year at most when we're taking imported water. So it might be a rental system we would bring in and rent it and use it and then have it hauled off, or maybe a permanent system depending on how that works out. But that's kind of the approach we've kind of discussed internally.
Great, that's really informative. I think we have time for one more question, and I think I'll ask this one before we close out the recording. So Kelly, we followed your adventures from sampling site to sampling site, and COVID definitely affected this study in very serious ways, but even more than what, in ways that were beyond what Kelly had to go through. Could each of you, Caesar as well, describe some of the unforeseen challenges that you had with this project? And I think I'll ask Kelly to start, because you had some very significant challenges that you overcame and did a great job doing that as well.
Well, COVID was a big one. I mean, the sampling event in 2019, we were excited to move on to Lake Matthews and Weymouth the next year, and hopefully get in another sampling event, and look at seasonality, which we ended up doing, but it was a bit of a stretch there. And in that stretch, I was pregnant and had a little girl. So she came along with me the second season with my husband. And so we made my 3-month-old do some science work. So, but everybody's been really great and understanding, and it went off really quite well. Another thing, I guess, to hit on that we found was we did have to resample Lake Matthews in the beginning in 2020 again, because we didn't sample deep enough within the water column. And so we only had 50 feet rope, and and got very small veligers and not very viable veligers, and it was super hot. So the next time we got 100-foot rope and we're able to go down further and get more viable veligers and do successful sampling. So that was my adventures for this study.
Well, congratulations on your new baby. That's terrific.
Thank you.
Adam, Caesar, anything to add?
I just want to say thank to Metropolitan and all the hard work you guys did to support us. I know COVID was a challenge for you guys as well. And so really appreciate you working with us to allow us access to these different locations during the lockdown. And of course, Kelly overcame a lot of obstacles. I think as with any scientific study, getting more data is always useful. And so I think in a way it was a blessing that we had to sample 2 years in a row, because we did see some variability in the Veliger size class distribution. And that really did provide us some more good information for this study that we wouldn't have had with only one sampling event. So in the end, it really worked out well for the study and for moving this forward.
Great. Caesar, anything to add?
Yeah, just echoing what Adam's saying, thanking Matt and Kelly's team over there and being patient, that 18-month COVID right out of the gate, we didn't know what we're doing day to day, let alone month to month. Many variables running behind the scene administratively. 18 months can push out a contract and trying to keep within the constraints of the contract. Thank you, Matt, for being patient and working with us on that. And great job.
You're welcome. Why don't we end it at that, unless, Don, you have any other burning questions?
No, not at this time.
Okay, well, this is a great presentation, a great recording. I want to thank Caesar, Adam, and Kelly for doing a great job on this presentation, but also managing the study. They're just barely touching the surface of the challenges that COVID had and the challenges we had with the agreements because of that. And so you guys, we were just the funding partner. You guys did the implementation, and you did the heavy lifting and the hard work there. And you shepherded through this COVID-19 crisis very professionally. And we appreciate that and feel that, this is a great study. So I want to thank you for that. I also want to thank the Metropolitan team. Don was the study lead for Metropolitan on this study and did a terrific job sort of shepherding it through this as well. Other folks on the team include Matt Hacker, Ariba Saeed, Stephanie Franco, who's on the call today. Teresa Gonzalez, Ryan Roman, and Lauren Carrillo have been helping from External Affairs and IT. And I also want to thank Hannah Ake, who helped organize this recording, but then left the district, but she was a critical part of the team, and she will be missed. This presentation is going to be on the webinar. And at this point, I think we're done with the recording. And once again, thank you. And why don't we hit stop record?